Intro music fades out.
You know, every single bioinformatics analysis has to start somewhere. And honestly, most of the time, that somewhere is the National Center for Biotechnology Information, or as we all call it, NCBI. It is, without a doubt, the world's largest library of biological data, and today we're going to take a beginner's tour.
So what is this thing? This massive digital library of life? Well, let's get into it.
Okay, so NCBI was started way back in 1988. It's a part of the U.S. National Institutes of Health, the NIH, and its whole mission is to collect all this biomedical info and share it. But here's the kicker. They made it all completely free for everyone. And that one decision, it totally supercharged biological research all over the world. So it's not really just a database, it's more like a global public library for all of biology.
And when I say library, I mean the scale is it's staggering. We're talking about data from over 260,000 different species. I mean, just think about that for a second. From the teeniest little bacteria to the biggest blue whales, it's all in there.
And if you think that's a lot, wait till you hear this. If we look at the raw data, the numbers just get, well, they get mind-boggling. We're talking over 100 billion. That's with the B. And that's just the number of DNA building blocks, nucleotides, stored in just one of its main databases, GenBank. What's crazy is that this mountain of data is doubling about every year and a half. It's an absolutely incredible resource.
All right, so a library this massive, where do you even start? How do you find anything? Well, you start by learning where the main sections are, or in our case, the key databases.
Look, NCBI has hundreds of databases for super specific things. But for most of us, your journey is going to start with just a handful of the big ones. First up, you've got GenBank. That's the gigantic archive for pretty much all DNA and RNA sequences. Then there's RefSeq, and we'll talk more about that in just a sec. PubMed, that's your gateway to millions of scientific papers. Then you have the gene and protein databases, which, you guessed it, do exactly what they say. And finally, SRA, the Sequence Read Archive. That's where you find the raw, messy data that comes right off the sequencing machines.
Okay, let's pause here for a second, because this is a really important distinction that trips up a ton of beginners. GenBank versus RefSeq. So what's the deal? Think of GenBank like this. It's a massive public archive where any researcher can submit their sequence data. It's got everything, which is great, but that also means it's super redundant and, yeah, it can have some errors. RefSeq, on the other hand, is the curated version. This is where NCBI staff actually go through the data and create a single, high-quality, gold standard record for a gene or protein. So the main thing to remember is this. GenBank is the raw, unfiltered archive. RefSeq is the polished, authoritative reference. Got it? Good.
And then, of course, you've got PubMed. This is basically NCBI's superpowered search engine for the Medline database, which means you get access to literally millions of scientific papers. And this is what's so cool about NCBI's whole philosophy. The data, like a DNA sequence, is directly linked to the papers that describe it. It creates this amazing interconnected web of information.
All right, enough theory. We know what's in the library, but how do we actually find what we're looking for? Let's do this. I'm going to walk you through your very first search using NCBI's incredibly powerful search engine, which is called Entree. Seriously, it could not be easier. First, just go to the NCBI homepage. You'll see a drop-down menu right at the top. You can pick a specific database if you want, like GenBank or PubMed, or you can just leave it on all databases to search everything. Then, just type in what you're looking for. Let's use the human BRCA1 gene as our example, and just hit search.
And boom! Look at this! The real magic of Entrez is that it searches all those major databases at the same time. So for that one search, for that one gene, you instantly get results for the nucleotide sequences, for the protein records it codes for, and even links to books and scientific articles about it. See how everything is organized and linked together? It lets you just jump from a gene to its protein to the paper that first discovered it. It's awesome.
Okay, so you found a record. Now what? You have to actually understand what you're looking at, right? So let's zoom in and learn how to read a standard GenBank file. And trust me, this is an absolutely core skill for anyone who wants to do bioinformatics. Every single GenBank record follows the same standard format. It's kind of like a library card for a gene. The locus line at the top, that's just a quick summary. Definition gives you a title in plain English. Now this next one is super important, the accession number. That is the unique, permanent ID for that sequence. It's like its serial number. It will never change. Then you get to the features section. This is where the real biological story is. It marks out where the genes are, the coding sequences, all the good stuff. And finally, you have origin. And that's just the raw sequence data itself. All the A's, T's, C's, and G's.
But you have to remember, finding a record like this is never the end of the story. It's just one little piece of the puzzle. It's one stop in NCBI's huge, connected ecosystem of data and tools. And honestly, this is where things get really, really cool. From any sequence record, you can directly launch powerful analysis tools. So with just one click, you can fire up the most famous tool of them all, BLAST. And BLAST lets you take that sequence and basically ask the entire database a really simple but incredibly powerful question. What else in the entire known biological universe looks like this? This is how you find related genes, figure out evolutionary relationships, and start to uncover what a brand new sequence might actually do. It is all connected.
And that brings us full circle right back to where we started. It doesn't matter if you're a student just starting out, a clinician in a hospital, or a seasoned researcher, your journey into biological data is almost definitely going to start right here. NCBI really truly is where every single bioinformatics analysis begins.
And that's your bite-sized tour of NCBI. If you found this helpful, definitely make sure you're subscribed and join us next time as we jump from the world of DNA into the major protein databases. We're talking UniProt, SwissProt, and Tremble. See you on BioinfoBytes.